Diffusion.
Hello,
Hello, how are you?
I'm good. How are you?
Yeah, good, thanks. Yeah, right
Uh,
in the middle of grant writing season, but other than that, yeah. So just Dr. Renee Goreham, a senior lecturer at the University of Newcastle.
so basically you're doing research into nanoparticles and you're focusing on applications in medicine for diagnostics and for treatment.
Yeah. I've sort of got two different areas of my research. I quite like them because it does span across different sciences. But yeah, the main one is looking at nanomaterials, specifically nature's own nanoparticles, which we call extracellular vesicles, but using these as a diagnostic tool. So determining whether they've come from a cancerous cell or a cancerous tumor, and then helping us to detect cancer as one of the applications. Yeah.
Right.
Mm.
And so how would you describe nanoparticles for the general audience?
Yeah, it's quite good. I like to describe these. I was doing it to year 12-year-old students the other day, and but yeah. So the way that I would describe a nanoparticle is, if you imagine your hair, you can just see the hair. It's a micron size, so it's still quite large. If you chop up that hair another 1 million times, then you'll get to the size of a nanoparticle, or something that I would synthesize, for example, like a gold nanoparticle. So they're really, really small, and they're so small we can't see them with our eyes or with a light microscope. But because they're so small, they have these interesting properties, and that's where I guess my sort of drawn nature towards them has grown from, is the fact that they're not what you expect, yeah, and what you would see every day.
So what sort of useful properties have you been able to apply?
Yeah, my favorites, they're called quantum dots. So as the name suggests, their physical properties are due to the quantum nature of them, and that's due to their nano size. So we can change their size from just being 5 nanometers to 6, so that 1 nanometer in change will change the color that they fluoresce. So each of these will fluoresce or be like a glow stick that we can then use. So, and just by simply changing the size. So they're already being used in QLED TVs. And so the application I use them in is I put something that targets something that's cancerous as another mode to look at cancer diagnosis.
And how did you come into using nanoparticles and nanotechnology?
I think that would be going back to the start of my career. So I did a Bachelor of Science, and then I went into nanomaterials in my honours year, but I was using like a nano-sized film. And then once I started doing my PhD, I started synthesizing gold nanoparticles. And that's another one where it has interesting properties. You think of a gold nanoparticle and you instantly think of jewelry. But when you actually see them in solution, they almost look like something you can drink, a nice red wine or some cordial. And that's again due to their nano-sized nature. So I was able to synthesize, or I was making these gold nanoparticles all throughout my PhD, and I was growing cells on to look at how the cells would grow on them, like whether they would like that nano-sized regime or not. So yeah, ever since then, I think I've just been drawn to the small natural, just small things, natural nanoparticles in general. So yeah.
And so you've been putting together the biological and the physical from an early part of your career as well.
Yeah. I don't have much background in biology, but I definitely know more than the average person now, and that's just through experience. But I have a lot of really great collaborators, and I guess that's one of the reasons I like this area, because I'm not just stuck in my little physics box. I talk to people all the time in chemistry and in biology, and we've created a nice little diverse group of collaborators.
Fantastic. So it's such an interesting area. I know that there's been research with gold nanoparticles for a long time and potential medical applications. Are there any in use at the moment in hospitals?
Yeah, I think there is. I haven't used gold nanoparticles for a while, but I believe their heating ability is used for to kill tumor cells. So you can heat them with a laser, and it will kill them. But also they're you can make them quite biocompatible, meaning they don't harm you when you get them in your body. And they come out of your body really easily. So there's no harmful effects when you get introduced them. So I don't know if there is, but there's been a lot of research in the past on drug delivery of gold nanoparticles and other nanoparticles as well. Even silver nanoparticles are being made, and they've got huge antimicrobial ability. So they've been used in filtration devices, and even bandages for wounds to prevent any infection from bacteria.
So how would you apply some of these nanoparticle properties to things like diagnosis?
Yeah, that's where I've changed from making my own. I still make my own. Well, I say me, my research group still makes their own nanoparticles. We're always making quantum dots. But then I found out, I think this is just after I completed my PhD, that our body actually makes our own nanoparticles, and they're really quite stable. These little vesicles that are nano-sized, about 50 nanometers. So different to my quantum dots, which are 5, but they're actually found in your blood, in your urine, and they're like a little mini-me of the cell. So the cell they came from, you can take this this nanoparticle, and it'll tell you what sort of state it is in. And so that's where I was like, I like nanomaterials. Let's have a look at these nano-sized objects that we have in our own body. And then it was our research that found them in breath. So if people are doing all this research to use these naturally occurring nanoparticles for diagnosis, why can't we then use the ones that we've found in our breath as a diagnostic tool? And I guess that's where I've gone for a really blue sky research, where I'm now looking at a disease breathalyzer, just by simply breathing. Yeah.
So we're breathing out these little vesicles all the time.
Yeah. And I guess you don't really know. They're so small anyway, so you wouldn't know. But it was one of my master's students who read this paper, and there's specific proteins that are present that indicate that you might have these vesicles there, and the proteins found in breath had these matching proteins. So we were like, okay, let's see if we can get some images using an electron microscope, and there they were. So it's just been 5 or 6 years of research in that area now.
Amazing. And what sort of diseases do you think you'll be able to diagnose?
Well, that's a really good question. I don't think there's a limit at this point. So we've started with lung cancer just because that would be the most obvious one. But we want to do a lot of extensive work on the fundamental, like where these EVs are coming, these naturally occurring nanoparticles, where they're coming from in your breath. Are they just coming from your mouth, or are they coming from your lungs? So we're not too sure, but I would like to think that it wouldn't just be for cancer, but maybe there's an opportunity there for asthma diagnosis, even stress, because stress markers have been found in breath. So yeah, we're really looking at the different avenues. And then more recently, we've gone, because of the pandemic, we've gone, okay, what about viral infections? So yeah, I think the applications here could be quite a number of them. Yeah.
There are so many well, not respiratory, so many aerosol spread diseases. That must be most of the ones we would be worried about. An awful lot of them would be from the breath.
Yeah. And I think so if we can tune in and find out what exactly is in there, because it's quite a complex mixture. You have these volatiles that just sort of evaporate off, but then you've got the liquid phase of your breath that has all this other messy content. And it's trying to decipher all that messy content to then be like, okay, can this indicate whether this person has a particular disease or respiratory concern? Yeah, so that's where we've really I'm trying to bring in my biologist friends to be like, okay, so how can we find out where these come from and what we can detect from them?
So if they're too small to see, how do you measure them?
Yeah, that's a interesting question. This is what I love about them because you have to prove they're there, right? I can tell you that there's nanoparticles in breath. And I've said already that you can't view them with a light microscope. The light's not going to see them. So what we tend to do, or what I like to do, is use two different methods. Electron microscopy. So we get a beam of electrons, and you can either bounce the beam of electrons off top of the surface, or you can shine the electron beam in enough energy that it will go straight through the surface. And so then it can image whatever is there. So if it has electrons or some sort of atoms there, it will have some sort of contrast. So that's the main way, and that's how I've imaged them regularly. But because I like physical characteristics, I also use this thing where we use atomic force microscopy, which has a nano-sized tip, and it can raster across the surface, and it just matches the height, basically, is the easiest way. And so we can image that at a nanoscale just by looking with a nano-sized object to match the size that you're trying to image itself. So they're the two physical ways that we can image them or try and visualize them. And they are, as you expect, quite spherical, just like a little ball. Or we can just look at the bulk solution and see if we can count them using light scattering techniques, which just shines light at it, and then we look at how the light scatters off the particle, and that can actually tell us what sort of size we've got and how many there are.
Amazing. So
Mm.
these little vesicles, do we know if they have, well, do we know why the cells are putting them out?
Yeah, well, originally when they were discovered, I think this was in the 80s, maybe 70s. It's been a while since I've gone back that far. They were just thought to be trash. So that's what the biologist thought. They were just expelling all the unwanted content. It wasn't until the 90s that they found out that they actually have a strong communication role. So the cells release them to get to another cell, and they have some strong communication. So they've actually got a lot of different roles, and they're involved in all sorts. Cancer in itself. So this thing called mesenchymal to epithelial transition. So it's where a cell turns from like a skin hard cell to like a soft cell that can go through the bloodstream as part of cancer. So yeah, it was all these different roles that people started to find. And then you've also got the content. So what is in the package is quite useful too. So they've got some little small DNA strands, some RNA, all different proteins. I'm mainly focused on what proteins they have because they can essentially be quite unique to the cell they came from. And so if we can find one that's specific to cancer, for example, we can target that protein, and that's what we can use to capture them.
That's, wow, I had no idea they still had strands of DNA and RNA and things inside them.
Yeah. It's just every few years there's just more and more information on them, and that's, I like to go to the conferences where we just talk about these naturally occurring nanoparticles and going, wow, I did not know that they're involved in fertility as well.
Ah,
So yeah, there's a range
wow.
of roles they have. Mm.
Do you want to talk about the fertility?
I don't know too much, but
Okay.
there is a research group here that looks at their role in fertility. So actually the content, I can say a little bit, obviously not a little bit, but the content of the EVs changes depending on the health status of male. Yeah, so the semen EVs will change depending on if someone is healthy or not. I don't know about women though. I'm sure there's a role there
There
too, but
must
I
be
know
something.
there's a research group. Yeah. They seem to be involved everywhere.
And you've talked about the vesicles as being EV. What does the E part stand for?
Oh, yeah, I always do that. So we call them extracellular vesicles, and actually my physics colleagues constantly get confused thinking I'm talking about an EV car. Um, yeah, so we call them vesicles or EVs for short.
Oh, that's, I think it's clear for the context, hopefully.
Yeah. Yeah, a quick correction and we're all good to go. So, mm.
Do you think it's possible? How long do you think these vesicles stay in the air? Do you think they could be detected after a couple of hours in the air?
Yeah, we are definitely looking into this. We want to look at the stability, but I do know that they are quite stable. I did some research with a colleague who was looking at the extracellular vesicles, or EVs, in breast milk. Again, another application therein. And that when we looked into the research, they found that those particles in the milk actually lasted all the way to the intestines of babies. So they were that stable that they could be consumed and go all the way to the intestines before they were actually taken up in for the nutrients.
Mm.
So yeah, and there is a huge amount of research in their drug delivery capacity because they are so stable. I mean, the cells make them so they are stable enough to transport this information or whatever they want to do across the other side of the body or whatever it does. We're still
Yeah.
trying to find out. But yeah, all we know is that they're extremely stable, and they're not going to be broken down easily. But that's one thing that our group was going to look at is, you have these EVs in an aerosol form or whatever, how long do they last? Yeah.
I was just thinking with the COVID applications, whether you could either have a little sensor, somewhere near your face that constantly monitors whether you've got it or not, or whether you'd have sensors in buildings that told you it was somewhere around. That might be
Yeah.
cool.
That we had a very similar conversation with collaborators going, how do we visualize this technology if it was to work? And we could imagine it just being incorporated into air conditioners, in airports, for example, airplanes. And that way you could just not only find out if someone's got COVID, but you could monitor it in real time. Obviously, very blue sky, but that's what we do.
Well, it looks like that's one of the many, many possibilities for this sort of technology.
Yeah, yeah, definitely. Hmm.
And you're talking about the vesicles in the milk containing nutrients. So are we talking nanonutrients as well as micronutrients now?
Yeah, I probably exaggerated a bit there,
Ah.
but I'm sure there would be some form of use that they would use in. But yeah, maybe that's to be determined. But
More
yeah,
research needed.
yeah, more research needed. It is an area that is growing, which is good. But yeah, it's definitely, even in cow's milk, they're looking at the extracellular vesicles. It seems like any living cell um, will form these naturally occurring nanoparticles. So yeah, applications are endless.
Amazing stuff. And so if students wanted to get into this medical nanoparticle world that you're researching, you started life as a physicist rather than a medical researcher. So what should students do?
Yeah, I mean, I'm still in the physics discipline, so yeah, I'm sort of an outlier here, but I still have a lot of basis. I guess, yeah, I like to look at them as physical entities and even, how soft or hard they are. But if you wanted to get into EVs specifically, it might be useful to start off with some sort of science degree where you have maybe some biology and physics, which isn't always, they don't always get drawn together, do they? So yeah, you get a lot of chemistry and physics majors, but maybe not that combination. But biophysics is still quite a new area. So I would suggest maybe going into that area and getting the knowledge of how we can use maths and physics to simplify these entities, but then also the application and the mechanisms that are involved in the body. So having that combination, I think, would work.
And there are degrees in nanotechnology now as well.
Yes, yeah, Flinders University offers, that's where I was, a great nanotechnology degree. So yeah, that's sort of where my love for the field came about.
Is there anything I haven't asked you that we should be talking about?
Um, I mean, there is the area of bacteria
Mm. Oh, let's
because
please.
yeah, I mentioned that all these cells, every living cell almost that we've found releases these nanoparticles, and even bacteria ones. So that's another aspect of our research where we're applying a similar technology, but then going, oh, okay, why can't we use this to detect E. coli, for example?
Mm.
So yeah, and the rationale for that is, well, nano-sized objects are actually quite easy to capture And if you imagine every E. coli expelling many of these extra these nanoparticles, then that'd be easier to capture in a higher quantity. So yeah, that's the other aspect they're looking at, water quality.
Wow. So that's, yeah, water quality, and I guess maybe even inside the body.
Yeah, yeah, definitely. I think we're just starting simple and going with water first, or even surfaces. So
Mm.
I mean, there's a lot of need for better pathogen detection for food, as well as hospitals and surfaces where you don't want any pathogens growing freely. So
Yeah.
if anyone is interested in extracellular vesicles, but with the more physical application, you know, I'm always open to having discussions. If anyone knows academics well, they know that we like to talk about our research. So you can always find my email and contact me more about any information that you've heard. There's always more to tell to the story. Yeah, so I guess
And you'll
that's.
be giving a talk at the Frontiers of Science Forum in March.
Yes, I am. Yeah, my calendar's already filling up quite quick this year. So I'm looking forward to having a bit of a different audience where it's quite broad, where there's politicians, students, industry, and then some of my science colleagues. So I'm looking forward to that.
Well, Renee, thank you very much.
No worries. Thank you so much. It was really a pleasure to talk to you, Ian.